Thank you all for coming. It's a pleasure to be here at the Broad. It's all a bit intimidating coming to kind of the temple of genomics and talk about structure. But hopefully I'll sort of convince you by the end that structure is beginning to be sort of good enough that maybe we could do some biology with it as well. This talk will be almost completely orthogonal to Sergey's in terms of the content. There's very little co-evolution here. In fact, it sort of by design doesn't use any co-evolution. But that will be sort of part of the background that kind of sets the stage for this work. So just to make sure everybody's on the same page, what is protein folding? And the basic process, the physical process, right, has to do with these unstructured polypeptide chains that we call proteins, which through largely kind of self-assembly take on a well-defined three-dimensional conformation, a sort of a low-energy state that corresponds to a three-dimensional structure. And what we would like to do is sort of build a parallel computational process that goes from the sequence to that structure, so basically predict how these things fold. And proteins come in sort of all shapes and sizes. And I should say that for a significant fraction of at least a human proteome, proteins are either completely disordered or at least have large fractions of which that are disordered. And for the purposes of this talk, at least, this is going to be not something I'm going to talk about. This is sort of beyond the scope of this work. But it's definitely of interest, I think, sort of to the broader biological community. Experimental methods have existed for a long time to try to tackle this problem, particularly X-ray crystallography, and not much more recently, sort of cryo-EM has really been making sort of substantial breakthroughs. Nonetheless, the success rate does remain low, and the average cost for protein structure determination is probably on the order of tens of thousands of dollars. All of these estimates vary. People in this room, I'm sure, are very familiar with this sort of picture that shows, at least in the sequence size, our ability to acquire sequences have been increasing exponentially, and so the cost of sequencing has been dropping exponentially. If you look at the analogous picture on the protein side, or the protein structure side rather, it's very, very different, right? So at best, if you look at the growth of PDB structures, it's a quadratic, but maybe even more kind of linear picture in sort of more recent history. So there's kind of an ever-increasing gap, sort of an exponentially increasing gap between the number of structures that we have and the number of sequences. And that's something that I think we'd all like to close because insofar as structure determines function, this would be a useful thing to do. So speaking of which, I like to kind of motivate this problem in sort of two ways. First, by sort of talking about practical reasons for why one would like to solve protein structure. And then sort of afterwards, I'll talk a little bit more about kind of a more broader or longer-term vision for why I think structure is useful. So probably the most often given reason is rational drug design. And the idea here is that you have the structure of a protein. You could impose something like a steric surface on top of it. And then if you're a medicinal chemist, maybe you see a little pocket here that you could then design a small molecule that binds that pocket and then modulate the function of the protein. And I think that there were surveys done about sort of asking medicinal chemists whether they'd be willing to work on a protein target without having the structure. And I think the vast majority, something like 80% of medicinal chemists, they say, we really would like to have a structure before we could sort of tackle designing a protein that targets it. When this work actually came out, there was a discussion on Hacker News, and this question of why do we care about protein structure came up, and this user kind of listed a whole bunch of reasons, and I think these are all great sort of practical reasons for doing so. And my favorite is probably this one. So I think if we have the ability to predict protein structure sort of reliably, then that may allow us to get some handle on the effect of genetic variation in human populations and elsewhere. And so that to me is sort of a compelling use case for what structure prediction can do for us if it gets to the point where it's sort of reliable enough. So these are all practical reasons. Now, there's, I think, kind of more of a vision reason, and that is to kind of build up a bottom-up systems biology, to try to get at systems biology from a kind of structural perspective. So if you look at a bacterial cytoplasm, this is an MD simulation of a bacterial cytoplasm. What you at least hypothetically should see is something like a whole bunch of proteins sort of interacting and sort of jostling for space within that crowded environment. And my sort of perspective on this is that much of what we see in terms of biological phenomena is ultimately emergent from these molecular interactions. And so I think our only sort of real hope for a ground unified theory, or at least a ground unified model, if you may, of biological phenomena as something emergent from sort of more basic principles would have to be grounded in structure. And so to me, on the sort of 20 to 30-year timeline, this is why I think structure could really sort of revolutionize the way we approach biology. Okay, so now let me talk a bit about sort of existing approaches and how people have sort of historically tackled this problem, if not practically, then at least just sort of paradigmatically. the first thing kind of maybe all this is this notion of molecular dynamics, right? So the idea there is that you're going to take something like a sort of a Newtonian approximation or a classical approximation of sort of molecular mechanics and combine it with Monte Carlo sampling. And what that lets you do is you go from sort of an unstructured polypeptide change to the folded protein. If you look at a simulation this is from the Pandey group at Stanford, this sort of simulation takes very seriously the notion that you would like to sort of reconstruct or recapitulate the dynamical trajectory of a protein folding, right? So it's not only about getting the final structure of the protein, it's also about sort of folding it over time. And that's a very compelling thing to do, but for practical purposes at least, it's not really within the realm of sort of realistic computation for the vast majority of proteins. Typically, no one really uses MD to actually predict the structure. It is used in the context where one already has a structure and is trying to understand the dynamics or certain sort of flexibility aspects of that protein. Then MD is useful, but beyond that, it's not really quite there yet to actually be used for making predictions. The second kind of class of models, and what I would say sort of, at least up until recently, was kind of the workhorse of protein structure prediction is fragment assembly. And so the basic idea there is that you sort of build a library of small fragments that have strong sequence structure correlation. So they're chunks of proteins that seem to have sort of consistent properties consistent structural properties given the sequence. And then the idea is one goes through something akin to sort of an MCMC process where you're swapping in and out these fragments while minimizing an energy function, as sort of an empirical score that one believes reflects sort of the energetics of protein structure. And then in a sampling, the hope is that, and with a good enough library, the hope is that one is able to sort of recapitulate or reconstruct the structure. In practice, this sort of historically only worked when you have an experimental template that looks a lot like your structure of interest. And you sort of work to sort of refine this template to give you the final structure. And so this sort of historically didn't really work very well sort of in a de novo way, where you don't have any information about the structure. In the last maybe 7 or 8 years is what I've sort of really kind of characterized as a revolution in the field, this sort of co-evolution evolution, which Sergey kind of touched on quite a bit earlier on, where one uses a multiple sequence alignment to infer effectively kind of geometric couplings based on evolutionary couplings, and then try to recover the structure that way. And often this is used sort of in conjunction with fragment assembly. Basically, these restraints sort of inform how this process is carried out to give you the final structure. I won't talk too much more about co-evolution because Sergey covered it, but let me just touch upon one aspect of it. So we have this sort of co-evolutionary picture, and then again, the idea is that you end up with this sort of contact map that is either used to sort of template match, so sort of pick out structures from the PDB that look like the same, and this is an approach that actually Sergey pioneered, or alternative, like I said, as restraints in a Venusia simulation. Now, the number of sequences tends to be quite large. I think it's gotten a fair bit lower now, at least initially. You need quite a few sequences to actually do this reliably. But my understanding is that even when you do have a sort of fairly good contact map, the success rate is still not 100%. So going from the contact map to a folded structure is still a problem in its own right. The pros of this approach is that it does work without supervision, so you don't need sort of structural data to train it, which is great, and that's sort of independent of known faults. So so you're actually able to sort of go into new regions of protein space that that one hasn't crystallized. And so one of the kind of the early successes in this area was from the Sanders group was in the context of the membrane proteins, where structures are fairly unavailable in that area, but these sort of methods were actually quite successful because we had many sequences. The drawback is that this approach is in some ways defined fundamentally on the family or MSA level, so it sort of doesn't perhaps quite have the resolution at the individual sequence level. It's really telling you something about the family as a whole look like, but not what an individual sequence looks like. And with things like de novo sequences, sort of if you're interested in protein design, sort of de novo protein design, this obviously has less to say because evolution hasn't given us sequences to sort of crib off of. Now, if you're to look at the state of the field at the moment, and so one of the kind of leading approaches is this thing called ITAZR, where you have, if you look at the pipeline that ITAZR sort of performs, it's very complex, and these two sort of blowouts here are just these two individual boxes. So you have this sort of very, very complex sort of human-engineered system, and Rosetta is sort of a similar thing, where it's about 3 million lines of code, and this sort of human complexity also shows up in computational complexity, where making predictions, say in the context of sort of cats, which is a sort of biennial competition, is actually fairly expensive. It takes quite a bit of computation to generate a single structure. And I draw this picture to contrast it with, say, where things were in computer vision about 10 years ago, where similar to how protein structure is today, you have these sort of very complex human-designed pipelines with many pieces and filters, things like SIFT, that people sort of painstakingly developed over decades. And obviously what's happened in the last 7 years or so is all of this has been kind of swept away by deep learning. Like that sort of come to kind of replace these very complex human-designed pipelines with at least sort of a higher level of human engineering and sort of significant simplification in terms of the underlying kind of computational systems. And so the kind of the motivation for this work was this question of whether the same kind of paradigm could be brought to bear onto the problem of protein foldings. Can one sort of bring this idea of end-to-end differentiability for protein folding and sort of do for protein structure prediction what's been done for computer vision and natural language processing and elsewhere? So the basic idea is to build a sequence-to-structure model where one takes sequence as input and spits out structure as output. So before I get into that, let me just give a little bit of really protein geometry just to kind of set the stage, because it will be helpful downstream. So this is what a protein backbone looks like. Each triplet here is a residue, is an amino acid. The nitrogens are in blue, and the carbons are in gray. And you can sort of have almost like a coordinate system based on this labeling. I'm going to sort of forget about the side chains for now and just focus on these three backbone atoms. Within these three, there's effectively sort of three potential degrees of freedom. There's bond length, so how far apart are two atoms from one another. There's bond angle, so if you look at three consecutive atoms and look at the plane of that, look at the resulting plane, then the rotation within that plane is another potential degree of freedom. And then finally, if you have the torsional angles where you look at four consecutive atoms, they form two planes, and then there's a sort of a hinge between those two planes, and the rotation along that hinge is yet another potential degree of freedom. So it just so happens that for proteins, you could effectively, for the most part, treat bond length and angle as fixed quantities. They're not really fixed, but you can sort of rejig them so that they do become fixed, and then the only remaining degrees of freedom are torsion angles. And for a way to do this, then, for a given residue, you end up with 3 degrees of freedom, the phi, psi, and the omega angles, and they're sort of well-known enough that they have their own names. And it's these angles that effectively kind of fully determine the conformational flexibility of the backbone of a protein, at least. And so these are going to sort of play a central role in sort of how one models this problem. Just so, again, by way of background, you may have seen the Ramachandran plot, which sort of looks at the empirical density of PDB structures as a function of the phi and psi angles. And it looks like this, where you have your familiar alpha helices and beta sheets. And again, this is just a sort of the kind of empirical density as we know it. It's worth noting that even though it's drawn as a sheet, fundamentally, topologically, its object is actually a torus. It's really sort of a 3 torus if you consider the 3 torsion angles. And so if one is trying to kind of model angles over the space, it makes sense to kind of take the topology seriously, although I won't really be doing this, at least for the purposes of this talk. But just sort of keep that in mind. OK, so the first kind of key building block that I'm going to use to build this scheme is a recurrent neural network. I expect most people know what a neural network is, but in case you don't, just the basic idea is they have some sort of some input in green, some computational units. They're doing some sort of computation that is learned. So this is the file that gets optimized. And then you have some output that you're trying to sort of get to in pink. This is the sort of standard neural network. In a recurrent neural network, these computational units feed back onto themselves, so they have a state that's sort of maintained as the computation is ongoing. And in particular, you can sort of imagine sort of unrolling that computation over time, where, for example, you have different amino acids, so you have a sequence of tokens, and then as you're proceeding through that sequence, information is flowing into these computational units from the inputs, as well as from the prior state of these computations. And the particular sort of kind of units that I'm using in this talk is what I call LCMs. The details are not so critical. I'm not going to go. I don't have time to go through this, but the basic idea is that you have these sort of gating mechanisms that control the information flow in and out of these units. And in particular, in this context, predictions are going to be made actually at every time step. So it's just a single prediction at the end, but rather at every residue position, there's going to be some prediction made, and I'll describe what that prediction is. And furthermore, the topology of the network is sort of bidirectional, so you're effectively going from the N to the C terminus and from the C to the N terminus and sort of sweeping across the protein in both directions. Okay, so I'm going to just kind of simplify this representation and just by kind of collapsing it into those yellow nodes. And this is kind of the starting point for building what I'm calling a recurrent geometric network that now, so this is sort of all off the shelf, and then from here on, I'm going to sort of augment it with sort of pieces that are bespoke for the protein problem. So the first kind of key piece is this one, having to do with kind of predicting angles at each residue position. So let's come back to this Ramachandran space picture, and one can sort of imagine this thought experiment where instead of trying to predict a structure from sequence, one can do sort of a sequence-to-sequence model. And one reason why one would perhaps want to do this is because there's a lot of work already in the natural language processing world in predicting sequences from sequences, for example, in translation. And so if one can sort of cast this problem as a sequence-to-sequence problem, it may be possible to sort of leverage all that machinery without doing too much work. So how would one go about this? Well, one possibility is you could take a Ramachandran space and sort of cluster it, effectively kind of tessellating into little chunks, and then collapsing each cluster into a single point. And what that does is that effectively discretizes the Ramachandran space. And from that approach, one can then sort of take a structure and then turn it into a new sequence, now not of amino acids, but rather of discrete choices of triplets of angles, of these points in the Ramachandran space. Okay? So just to be clear, there's no machine learning here so far. All I'm saying is I'm going to take a structure, and I'm going to sort of replace it, which is effectively a continuous curve in 1D space, or a continuous 1D curve in 3D space, rather. I'm going to take that structure and replace it by this discrete sequence of tokens, right? And these tokens come from this discretized Ramachandran space. Now, this doesn't really quite work, and I'll explain why. So what I'm showing here is you could vary the alphabet size. You could vary how many discrete angles you consider, and look at the RMSD of the resulting sort of structure that's resultant from this sequence for at least a small window, so just 7 residues at a time. And what you see is that there's a fair bit of error even for fairly large alphabet sizes, and it sort of saturates that. It doesn't matter how big your alphabet size gets, you're stuck with that error. And this is true even if you sort of flip the picture where you change the window size and fix the alphabet. As you increase the window, your error starts sort of going up, and it just eventually kind of degenerates. And the reason why this happens can be explained fairly intuitively. So you imagine you have a sort of a single curve in 2D space now, and you have continuous angles at each position, right? So what I'm proposing to do, right, is we have these initial angles, and I'm going to replace them with these discrete angles coming from this fixed alphabet, right? And what that would do, this sort of approach, this kind of local reconstruction approach, is that locally the angles will be a reasonable match to the original structure. But globally, if you have this sort of ruler here, you see this accumulation of error that starts deviating further and further as you add more and more residues, and as the error accumulates further, more and more. So this is the reason why you're seeing this problem. And irrespective of how big your alphabet is, you sort of never quite, at least for real proteins, can never really quite reconstruct them in this approach. However, one can do something different. And instead of sort of greedily picking the closest match, one can instead assign alphabet choices globally in a way to sort of optimize the global fit irrespective of the local fit. And that would look something like this, right? Where locally, the angles are rather a poor match for the original sequence, but globally, there's not this deviation that one saw here, right? And so this is at least the idea that may work to sort of try to tackle this problem. And in fact, it does work. So in this case, using the sort of global reconstruction approach, as you increase the alphabet size, you see the RMSD for a much larger window size now actually starts decreasing and continues to decrease as you make the alphabet larger and larger. So that's interesting. Sorry, just one last point. Yeah, I mean, if you look at the kind of resultant sort of clusters of fragments that have to do with these kind of clustered sequence fragments, you see fairly tight structural kind of ensembles. Okay, so what this is suggesting is that with sort of local reconstruction, we really need sort of an effective alphabet size of infinity. But with global reconstruction, we could get away with potentially just two kind of discrete angles to reconstruct any shape if we don't care about local quality. There is a problem though, which is global reconstruction leads to a sort of a sequence indeterminacy. And what I mean by this is that if you take the structure, you could have one particular sequence that gives you, reasonable accuracy. But you could have other sequences that are actually quite comparable in accuracy. And in fact, I think it's either an NP-complete or NP-hard to actually sort of figure out the optimal letter assignment that sort of minimizes the structural match. So in general, you're doing some sort of heuristic anyway to get these sequences, and depending on that heuristic, you could end up with different sequences depending on sort of the random initialization or what have you. And this becomes a problem now in the learning context, right? So if one wants to formulate this as a machine learning problem, because you have to sort of induce an invariant structure that understands how all these various sequences degenerately map onto a single structure. And that becomes very difficult. And in fact, I actually tried to get this sort of approach to work for quite a while, and I could never really get it to work, I think in part because of this reason. Okay, so that doesn't quite work, but that sets the stage for what does work, which is what I'm going to explain next. So previously we said we'll have discrete outputs and something like a sequence loss. What I'm going to describe now is actually a way to sort of formulate this notion of an alphabet in a continuous manner, and which is useful because it gets around the problem I just described, but it also allows us to do something more sophisticated downstream, which is quite nice. But let me first talk about this continuous idea. So we're going to sort of continuously relax the notion of an alphabet. And this will have the advantage of allowing us to predict sort of an infinite alphabet size in practice and maintain differentiability, which, like I said, which I alluded to, sort of allows us to do kind of more complex losses downstream. All right, so we'll still have an alphabet, and I'm going to represent it by sort of these capital thetas. And so now this is the machine learning context. Now it's not just simply a lookup. What we're going to do at each time step is predict a probability distribution over that discrete alphabet. So it's effectively just a softmax. In this context, though, I don't want to sample from this distribution. I actually want a single number. So I want to deterministically predict a structure. And so I have to somehow take this information, this sort of probabilistic information, and sort of extract a single prediction out of it. And the way I'm going to do this is, so this should kind of make you a little bit queasy, because what I have here are a bunch of angles, right? And so you have a bunch of points on a circle, right? And I want to sort of average these. And that's not a particularly well-defined concept. Whoa, what just happened? Yeah. Oh, okay. So one has to somehow collapse these sort of angles onto a single angle and sort of average them in a certain way. So there are different tricks of doing this. One trick that I use is the following. I treat these points as if they were lying in Cartesian space. Right? So they're angles, they're not vectors, but I treat them as if they were unit vectors. And then I weigh them by the associated probability coming from the softmax. And I simply average these vectors in Cartesian space, and then sort of pop it back to a unit vector to extract the angular information. So I'm sort of doing this kind of weird trick where I start out with angles, move to Cartesian space, and then extract an angle out. But for the most part, this works quite well, and this allows us to sort of effectively extract a single number out of this probabilistic picture. Right. So the first step now is that this is going to allow us to predict triplets of angles, torsional angles, at each time step. But that's not enough because, well, one can imagine doing things in sort of angle space, but that ends up having its own sort of issues. What is nice now, because this is all differentiable, is we can actually sort of push this forward and predict the actual structure in Cartesian space, the actual atomic coordinates of the structure. And the way I do this is fairly simple. The angles are sort of fed through these units that deterministically reconstruct the structure of the protein. And I'll also just kind of briefly walk you through this. So at each time step, 3 angles are being predicted, and then the previous atoms that have been constructed up to this point are sort of fed. Based on the torsional angle, there's a rotation that's made that's predicted. And there's a bond rotation that happens that's not predicted, that's just fixed. And then there's a bond extension that occurs that's also fixed and not predicted. And then this process is just repeated over and over again, effectively kind of sequentially reconstructing the structure. And in fact, if I let this animation run for 25 minutes or so, you'll actually see the entire protein gets reconstructed. In practice, I actually don't do it sequentially like this. I sort of, in parallel, generate separate fragments and then put them together at the end for computational reasons. But mathematically, it's the exact same process. And this is sort of the underlying equations. But the point here is that there's nothing learned here. This is all purely deterministic. And the only point of this is just to recover the structure so that then one can do sort of optimization directly in the global structure space as opposed to an angle space. All right, so the final step now is that we have the predicted structure, we have the experimental structure, and we need some sort of loss function that measures how close the predictions are to the ground truth and use it as an optimization signal to learn a model. So you can imagine that if you have sort of two structures that are kind of in the same arrangement, that this may be kind of a trivial problem. But at a minimum, we know that structures are sort of invariant up to a rigid transformation. So we don't quite know what the general position and orientation of the predicted structure versus the real one. And so whatever loss metric one has to have should account for this. The other aspect, or the other important piece here is that, at least initially, predictions are likely to be very poor. And so we would like to have some way of assigning partial credit so that the model is able to sort of pick up on local agreement, even if globally the structure is quite poor. And the reason I say this is because there are approaches which sort of force an alignment of the two structures before a metric is computed. And that, I think, is somewhat problematic in this context, because if you were to force an alignment, it would be hard to get partial credit. So the approach I take is the following. I take the predicted and the experimental structure. I compute all the interatomic distances within each structure independently, which results in a distance matrix for each structure. And this distance matrix is invariant to translation and rotation because it's just defined in terms of distances. And then I take the distance between distances, basically, of these two matrices, and then just sum everything, or average everything. And that results in a single interpretable number that corresponds to something like what's called a distance-based RMSD. So the advantage of this is that it's differentiable. It's alignment independent, so you don't have to put the two things together. It is, on the other hand, size sensitive. This is a drawback, so it tends to kind of grow with the size of the protein. And it is invariant to reflections, so it doesn't quite know about chirality, which is also sort of a drawback. There is an alternative score, which is widely used as well, called TM score. At least in the sort of original formulation, it's not differentiable. But the bigger issue is that it's alignment independent, so it does force an alignment, which to me is a drawback. On the other hand, it has better properties in terms of being size insensitive, and it is chiral, which is good. So I use this as a way to kind of assess the model, but I don't use this as a training signal. One last bit of sort of modeling before I go on to the results. So I said I feed the amino acid sequence. That's not entirely true. I feed it something a little bit more. So I do build a multiple sequence alignment, but then I collapse it onto a position-specific scoring matrix. So every position gets a probability distribution over the likely amino acid propensities. And so you can think of the PSM data complexity as something like ON, right? So you need some constant factor of n quantities. You need to estimate that many quantities, that many parameters. While in the co-evolutionary context, it's really more like n squared, because you're trying to sort of estimate all the couplings between every residue. So in principle, at least, this may sort of work better in sort of very low data regimes where you don't have enough sequences to reliably estimate something like n squared numbers. All right, so let me discuss the results now. There is actually sort of a bit of a challenge in sort of assessing these sort of models. And part of the reason is because new protein folds are a little bit hard to sort of to define. It's a bit of a subjective question whether this is a new protein fold or not. But you would like to do that for the test set, to sort of subject the model to a difficult challenge, right? So thankfully, there's this thing called CASP that happens every 2 years, where new protein structures that have been determined but that have not been publicly released in the PDB are given to predictors, the sequences of which are given to predictors to predict blindly. And then at the end of the year, the structures are compared and the winners are announced. So the basic idea was to kind of piggyback on CASP, where the test sets are the CASP sets, and then the training sets correspond to everything that exists prior to a given CASP, all the sequences, all the structures that are available prior to a given CASP. And another nice thing is that CASP organizers kind of delineate structures into two categories, into TBM, template-based, and free modeling-based, corresponding to sort of roughly known or novel folds. And so that kind of provides a third-party sort of objective characterization of the difficulty of protein targets that I use to kind of break down my predictions as well. So let me start out with some representative prediction just to give you a sense of how well this works or doesn't work. So this is what I would call kind of a good prediction, experiment on the left, prediction on the right. The prediction is about 2.5 angstroms in resolution. The diameter of the protein here is about 40 angstroms, so that gives you a sense of the scale. A hydrogen bond is about 1.5 to 2.5 angstroms, so again, that gives you a sense of scale. For TM scores, I should say, they range from 0 to 1, 1 being best, 0 being worst. And a score below 0.17 is typically considered random, and about 0.5 is generally the same fold. So this is, in general, this is a good prediction. This is what I would call sort of a fair prediction. You see this sort of helical region here is well predicted, but the C-terminus is largely unfolded. There's actually more going on here, though, is that because even in this well-predicted region, the secondary structural element, if you pay close attention, are overly wound in the predicted structure. They sort of the turns are too tight. And it turns out that basically the model sort of learned to do sort of effectively non-physical torsion angles to get this sort of thing. So it gets the structure globally correct, but locally not quite correct. And sort of harkening back almost to the discussion I had earlier about this alphabetization business. And then finally, this is what I would call a very bad prediction. There's very little agreement between the prediction and the ground truth. And that's reflected in the RMSD and the TM scores. I would say that for most known false predictions, the accuracy falls between good and fair, and for most novel falls, it's between fair and poor, something on that order. But I'll give you some systematic numbers now. So for FM category, these are the novel false prediction. I'm showing here for the last 6 casts, except the very last one that just happened. The top row is this model, and these are the top 5 servers at each given cast. And so here it's doing quite well. I wouldn't necessarily, the take home here is not that it's the best, but that it's competitive with the best. There's some technical things here I'll skip. But yeah, so this is the RMSD. If you look at TM score, it's usually in the top 5, but not necessarily the top 1. So this is why I was saying it's sort of competitive with the state of the art, but not necessarily the best one, really. It varies, obviously, by structure. If you're looking at known false, so this is where it's template-based, the situation is different. It's not quite competitive yet with these approaches. But the caveat is that these methods have access to an experimental template, so they usually have something that looks a lot like the thing you're trying to predict, while this approach is sort of working in a de novo way. And this is also reflected in the fact that these approaches typically sort of have sort of 2 separate pipelines, one for de novo prediction and one for template-based, while in my case, it's a single model that's predicting everything at once. And same story with the TM scores, where there is a sizable gap. Although in this case, like I said, this is sort of around the 0.5 kind of threshold. One point I'd like to address. So there's a question of, when I'm saying sort of not template-based, it is obviously trained on the PDB. So in some ways, it's sort of learning from data. So in what sense is it not template-based? And at least in one particular version, it's not, and that I'll describe next. So you could do this experiment where you could look. So the CASP organizers, right? They obtain templates for the structures that they know about, because they have the structures that look very similar to the structures. And so what one can do is ask, what is the RMSD of the template with respect to the true structure, and compare that on the y-axis, and compare that to the RMSD of the best prediction at a given CASP to the true structure. And I'm doing this for small windows here. So for windows of 5 residues, I'm looking at various casts, at the correlation between the template and the best prediction. And what you see is that it's a reasonable correlation. And this correlation holds at different length scales, suggesting that the methods work well when there are good templates, and don't work well when there are no good templates. The situation is quite different for the RGN model, where the correlation is gradually 0, irrespective of how long your window size is, suggesting that whether the RGN works well or not, it's sort of independent of the availability of a good global template. And so that's, at least in this sense, for kind of downstream biological applications, I think it's potentially sort of a useful thing to have. Just for fun, I'm showing here in white an experimental structure, and then in color a predictive structure as a function of model training, right? So as the model trains, initially sort of comes up with this sort of slinky-like thing, and then as it gets better and better, the structure starts settling down onto kind of the final thing. I'm not sort of claiming this in any way mirrors the folding process. This is really kind of more of a reflection of the linear dynamics of the model, but it's sort of kind of intriguing to look at nonetheless. Okay, so so I have about 10 more minutes. I'm primarily going to talk about sort of some of the implications of what I think this sort of approach is, and also about what's happened in the last year or so, because it's been a fairly active and exciting year in the space. But let me first talk about kind of the implications. So there's no conformational sampling, no energy minimization. There's none of the kind of conventional things that you do in protein structure prediction. And so the SPS are several orders of magnitude, right? So it's really the comparison between milliseconds versus days. And that's a fairly consequential thing for many applications. For example, if you're interested in things like docking, where you're trying to take a small molecule and fit it inside a protein, typically this is done in a rigid way where the protein is held fixed and the molecule is moved around. And that's at least in part because predicting a new structure every time a molecule is moved is very expensive. But if this can be done very rapidly, then one can imagine doing this instead of in a docking context. Protein design obviously is another kind of interesting area, and generative models we mentioned earlier. I think that's sort of coupling this with various generative approaches would be sort of a very compelling thing as well. Effects of genetic variance, that's another issue where I think things like co-evolution models are maybe not as effective because of this issue of being kind of MSA-based. And ideally, I think one would like to even get to the PSSMs and just have the prediction be done on an individual sequence, but we're still not quite there yet. And perhaps, sort of inside of these RGNs or these sort of models in general, there may be some interpretable insights about the nature of protein foldings that we can extract one day. And we can already see hints of that. So what I'm showing here is the kind of a representation of the latent space of the RGN, where each dot corresponds to a protein sequence. And this is a 2D projection of a sort of a 4,000 dimensional space that the RGN builds. And what I'm overlaying on top of that projection are the secondary structure contents of each protein. So what's interesting here is that obviously uses that information to sort of lay out its structure, its representation of protein sequence space. And this is interesting because secondary structure was not used as input, and it was not used as a training output either, as a training signal. So there was no explicit information whatsoever that secondary structure even exists. Yet the RGN learned to use it as sort of the primary way it organizes its sort of vision of protein space, which I find is really interesting. And I think so digging deeper into this is actually a fairly fertile ground for further work. So coming back to this kind of picture, to me this is sort of a scaffolding upon which one can build many things, right? So you have sort of a torsional local perspective here that can sort of easily incorporate things like secondary structure. One has a global sort of representation here, right, where you sort of the protein is in sort of full Cartesian instantiation, and one can imagine things like convolutional networks sort of operating on this representation. And then one has a sort of a distance-based kind of multi-scale representation in the RMSD loss, which can incorporate sort of distance-based kind of information, things like co-evolutionary data, or things like, say, physical priors like coulombic repulsion. So I think all these sort of pieces can be used to kind of plug in additional pieces of information that could potentially sort of, perhaps complicate the model, but also sort of make it more performant. Okay, so let me briefly talk a little bit about sort of last year, because it's been a fairly kind of exciting year. So this work was the pre-printed around February. In September, John Ingham gave a talk last year on this approach, this notion of a differentiable simulator, which is also an end-to-end differentiable approach. They're independently developed, and they're quite different in terms of the way they actually tackle the problem. But they share the same spirit in terms of trying to kind of cast this problem as an end-to-end differentiable problem. In November, in a completely different kind of space, in the kind of more the co-evolution space, so both of these are kind of trying to get at this problem without using co-evolution. Jimbo Zhu sort of put up a preprint on using kind of distance-based learning, so not predicting contacts, but predicting distances, which seems to be quite performant. And in fact, it sort of turned out to be kind of one of the key ingredients of sort of DeepMind's approach at Cas13, which really kind of put a sort of a sizable gap between them and kind of the closest competitor at the competition. So just to kind of try to lay these things out in kind of model space, I should say there are at least sort of 6 dimensions which are perfectly good to sort of lay out a model space. And I'm only looking at 2, and these are not in any way kind of the best 2, but it's just a way to look at this problem. So one can sort of imagine, from single sequence models to multiple sequence alignment models, co-evolution models, and from sort of very minimal kind of purely data-driven to sort of more complex physics-based approaches. So the RGN model I described sort of really kind of lays on this side of the picture, and it's not quite here yet because it's using PSSMs. I would like to get it to that level. The NEMO approach, so John Ingham's approach, sort of is maybe a bit more complex, has a bit more kind of physics in it, and it's sort of roughly kind of the same level in terms of the vertical axis. Something like Raptor X, the Jimbo Zhu's approach, these co-evolution approaches are very much in this category, right, so multiple sequence alignments. This particular model is fairly minimal. AlphaFold's approach has a bit more physics in it, but it's really not just about that. There's a lot more stuff going on. The machine learning here is a lot more sophisticated than say what's going on here, and that's partly, I think, what's driven their performance improvements. Something like Rosetta is all the way up here, kind of just pure Rosetta is kind of more physicsy based, and in principle at least works with a single sequence. And when you add things like Gremlin, like Sergey's stuff, you can bring it down here to allow it to sort of take advantage of co-evolutionary information. There's a question. So one point is to say there's a lot of space to sort of operate. There's a lot of interesting questions. And to me, at least, I think that maybe the holy grail is somewhere here. Perhaps not. The x-axis is somewhat irrelevant. I mean, anywhere here is interesting. But I think being able to sort of predict just from single sequences is what I think are the most interesting questions, particularly for downstream biological applications moving forward. Okay, so I think I have 5 more minutes. So I'll finish up with a question as opposed to then with a kind of a proposal. And the question is the following. So I'd like us to think a little bit more about sort of what it means to do theoretical science, kind of more broadly, and in particular in this context, right? So one version of what theoretical science is, right, is that one has sort of an observation or an idea that acts as a guiding principle, as a prism to interpret both data and to sort of do model building, right? So that's sort of one version of what a theoretical science is. And there have been many examples of this, things like maximum entropy principle, which actually has been kind of the underpinnings of co-evolution, but in other areas, say in physics, things like scale decouplings of symmetry that have showed up repeatedly and have led to sort of very important breakthroughs in the physical sciences, at least. Now, in the context in which one is sort of specifically not interested in high energy physics, right? I think that there are a couple of other sort of points that I'd like to add. One is that we would like to kind of mathematically describe a class of phenomena kind of sufficiently accurately to get their salient features, but to do so in a way that sort of abstracts away from the underlying physics, right? I mean, I think that the idea for us, as for sort of non-physicists, is to try to sort of build a theoretical model that kind of captures the phenomenon at a sort of in a way that sort of gets at what's interesting about that phenomenon specifically, independent of the underlying physics. And there are many examples of this, right? I mean, condensed matter physics doesn't go down to the underlying quarks, and things like organic chemistry and evolution certainly don't. So is there kind of an analogous picture for protein folding? So I'm going to sort of propose 3 observations that I think can sort of maybe allow us to sort of begin to build something akin to that. The first observation is this notion of folding by nucleation. So we know that there are these energy landscapes that proteins sort of move through to get to the lowest energy state. And at least from molecular dynamics simulations, what we've seen evidence for are these sort of processes in which local regions of the protein take on sort of a local conformation. They act as a nucleation site, which then in consequence, lead to formation of higher order structures. Right? So there's sort of a hierarchical process to the folding process, in which you start out from small regions and so go on to sort of higher and higher order regions. At least from MD simulations, there's evidence for that. That's one bit. The second bit is that there's sort of increasing evidence that early life was sort of a peptidic soup. So if you look at kind of the ancestral proteins, you find these sort of small regions, these small peptides that seem to sort of independently have either catalytic or structural properties, right? So, and that there's evidence that sort of today's proteins, the extant space of proteins, is sort of effectively kind of emergent out of combinations of those kind of ancestral peptides. And so there's kind of a sort of a history to the way that evolution sort of has kind of driven the course of the extant protein space. That's 2. The third piece is this notion from really more basic computer science theory about modularity and hierarchy and how they improve evolvability. This picture doesn't really mean anything, just a pretty picture, but the basic idea is that what we've seen in kind of computational evolution experiments is that if you have modular systems, these modular systems enable evolution at a faster rate. That the more modular a system becomes, in the sense that subparts are decoupled and they're sort of independently evolving, the more rapidly one is able to evolve. And so this suggests this as a mechanism, modularity meaning, as a mechanism for sort of meta-evolution, for sort of speeding up evolution. So taking these 3 pieces together, I think kind of gives rise to what I'm sort of calling a kind of a linguistic hypothesis. And these ideas have been around for a long time, that sort of something akin to natural language structure may underlie a protein sequence space, meaning that there are things like sort of fragments, like words, and there are certain sort of grammatical rules, certain kind of emergent heuristics that describe how these fragments are combined together, and that the course of how these things have arisen akin to natural language is very much driven by historical accidents. That sort of things happen at various points in time, and these locked in certain fragments, certain words, and that lock-in sort of drove kind of further evolution downstream of that. And why I think this is interesting is because everything I've talked about today, in this kind of deep learning context, has to do with formalizing a prior over the learning process, right? About how can we design efficient machines at extracting information from data? How can we make them learn well? But it's sort of a parallel question, which is how do we formalize the prior over the generative process, right? I think we have a lot that we already know about how protein sequence and structure space goes, and what's the best way that we can sort of formalize that prior knowledge over how proteins were generated? And that's an open question that I have some ideas, but I think it's something that the field it would be worthwhile for the field to think about. So with that, I'd like to sort of acknowledge I've had very many collaborators and people who've helped over the years. I'm very thankful for all the contributions. And there's a couple of papers. The main one is the top one right here, and a couple of technical follow-ups. There's a blog post, data is online, and code is online. And thanks for your attention.
for this thought-provoking presentation. Do you mind going back to the slide where you had of your, I guess, of your poor CASP model?
My poor CASP model?
Yeah.
This one.
Yeah. So there was, I thought I saw it. It looked like maybe it was a little overly wound up in some spots in the blue region there. So it looks like it's almost
like
like
overly wound. Everything's overly wound. Yeah.
So is there a way to fix that without using, say, like a van der Waals term or something more physics-based?
Yeah, I think so. So I didn't go into the technical details, but one issue here is that the way the penalty is constructed is that it's only penalizing on the C alpha carbons, not on any other backbones. So the backbones are reconstructed fully, but the penalty only looks at the C alpha for computational reasons. And I suspect that has to do a lot with this sort of overly wound local structure. So I think fixing that would take care of this problem. That's my suspicion, at least.
Related to that, actually, I wanted to ask, your penalty is uniform over all of the inner molecule distances, right? So an error of 2 angstroms at 40 angstroms distance is the same penalty as 2 angstrom error at 4 angstrom distance, so right?
That's right.
So the over 1, you could be trying to compensate for these long distance. Okay. Interesting. So potentially just, a different loss function based around, like, if something's 40 angstroms or 43 angstroms away, it's probably not as much of an error.
Yeah, absolutely. I mean, that makes a lot of sense to try to effectively say, if you're so far apart, it's sort of a lost cause anyway, so don't worry about pinning it down.
I was going to ask if you've done any experiments on weighting the loss function or other parts of that, and what they told you about the importance of these, or if this is the primary loss function.
Yeah, I have. I would say where that's played the biggest role in making things work better is when I try to push this to predict from single sequences. It's less critical if you're predicting from PSSMs, but if you're trying to actually make single sequence predictions, being much more careful with how the loss is constructed helps a lot, because I think the optimization landscape becomes much more difficult, and so the problem gets stuck in local minima all the time, and so those considerations become much more critical there. Yeah.
It's a great talk. I like your philosophy and your work in general, but I'm wondering, like, if you think that the folding is happening in hierarchical fashion, have you ever considered applying an attention mechanism so that, like, the generation will depend on certain regions, not doesn't have to consider the entirety of the sequence, because it's a common model that's being applied to sequence models, which is very similar to that.
Yeah, absolutely. I mean, that's what's so great in being kind of the NLP space in a way, is because there's all these developments that have been very exciting. So yeah, absolutely. I mean, I don't have any results to sort of report, but that's definitely something we're looking into. And I think the nice thing about sort of an attention-like mechanism is that immediately kind of provides interpretability for the model. So, I mentioned earlier about sort of trying to kind of crack open these things and try to understand what's going on inside. I think having an attention mechanism would potentially sort of immediately kind of show things like, this particular fragment likes to couple with this other fragment and so on. So I think that's a very exciting direction to go into, yeah.
Just related, so very good talk, I think. The one comment just came up, which is that none of us actually have used, I mean, like we have the EV fold server and, the work that Sergey talked about and others and so on, not to repeat. But, sequences have evolved not just to fold correctly, but also to avoid incorrect folding to some extent. I mean, there's some sequences, these are rare exceptions, which actually can exist in 2 conformations in a biologically meaningful way, and you get superimposition of the nuclear couplings and so on. But the general statement is that organisms that have proteins that misfold or don't have a high folding efficiency typically would get rid of those proteins. So the challenge is for all of us, I think, in this field to actually use that, perhaps. That's the question. Could we find a way of injecting that into our learning processes to actually use the fact that to get the correct protein sequences, don't just get the correct fold, but also avoid misfolding? It's not easy to do, but I think it's something we haven't used. Do you agree?
No, absolutely. And I think it's very interesting, especially in the protein design context, right? Because in some ways you could sort of almost pretend like this is not an issue in the natural proteins context, because you sort of, you know, if you're just learning from data, you can sort of stay along that manifold that looks like foldable space. But if you try to actually go away from that manifold into new protein space, truly new protein space, and that problem is going to be really serious.
I mean, there could be local misassociations, and one may be able to capture that. And when you describe some local properties, so I'm triggered partly by what you're talking about.
Yeah, that's interesting. I mean, you mentioned this because we've actually seen this with some of these design proteins, like from the Baker Lab, where they're, you know, locally they're very well. They look like protein space locally, but globally they don't. And the model is actually able to pick up on this in the sense that it sort of detects very tight alpha helices locally, but globally so it doesn't know what to do with it. So it's sort of an interesting.
Just one other comment while I have the floor, which is, you know, the question that just in the back, the one the sequence on the right. It's another challenge. I don't know if anybody has, maybe Sergey does, a good software for that. When we fold proteins using elusive couplings, you know, we sometimes see, you know, bad threading.
Yeah.
Things go under in a way that's sort of visually obvious, but there are no clashes. The van der Waals are okay, etc., etc. So one can do a space curve definition that quantifies knottedness or other incorrect topography. And I wonder whether somebody out there recently has developed a space curve thing that would allow us to just get rid of those.
Yeah. Add that as another loss item, effectively.
Do you know any?
I know historically people tried like filtering through decoys to remove anything that had dots in them. Just because but I don't think it's been used during.
Yeah. I mean, there are people, you know, in the old days, when I say, you know, for DNA supercoiling and so on, people have, done space curve integrals, and knot theory has done some of that. So there's mathematical theory out there we could use for that. It's not a totally new problem, but it hasn't been applied here, I think, yet, not yet.
Well, in some ways, I think it hasn't been applied probably because in some ways it's hard to use, except like Sergey said, like as a filter up to now. But I think with these sort of end-to-end differentiable approaches, you could just add as a new loss item, and then you can just feed it through. So I think now is the time in some ways to actually begin to apply these sort of things. Yeah.
Maybe with like a GAN approach where you have, you know, this doesn't, like, you can look at this, I mean, you say this visually doesn't look very good, and I'm like, I have to look at the labels to see which one is wrong. But, you know, if there are examples that are just obviously wrong, then maybe train a network just to distinguish, you know, real.
Okay, that might be a nice, simple way to do it.
Right, especially if you have a large database of like, this is what I came up with, and it's like, no, that's, you need to optimize for reasonableness and physical, sort of this behavior.
Yeah, that could work, yeah. Good idea, yeah. Supervised training.
Thanks. Thanks for your talk. I was wondering, can you account for like non-protein elements within the framework of the model you designed, or how would you generalize it to account for, say, a small molecule ligand or a DNA molecule?
Yeah, sure. So that is how could one generalize the model to sort of account for non-protein molecules like a small molecule or a DNA molecule? So one general area that I'm sort of excited about is kind of working with the latent space representation, because that's sort of a very rich thing that even if you sort of don't care about the structure, you kind of care about what the thing thinks of the sequence in structure space. And in that space, one can then imagine, at least, sort of, let's say you're sort of interested in the drug problem, right? So you can imagine sort of an encoding for the drug and encoding for the protein into the latent spaces. And then you can even potentially have some sort of geometric descriptors that relate them, and then doing the learning on that space. So that's at least one idea in which one can sort of imagine doing some version of this.
Yeah, I was curious about your procedure for averaging angles. I understand that, actually I want to ask, is the reason behind that procedure just to maintain differentiability, or any other intuition? Because physically it doesn't seem the right thing to do, right?
Well, so the averaging is being done in sort of in this thing's brain, right? So it's not sort of meant to kind of reflect the underlying physics. Obviously, it's sort of it's basically learning to predict angles so that when they're averaged, they give you the right answer in a way. So this is really kind of all in sort of machine learning space, if you may, or model space. But to answer your question, yeah, in a way it was to maintain differentiability so that we have a way to kind of extract information out without having to sample. Having said that, there's nothing really preventing one from, in fact, using this approach and just sampling from it. I mean, there are these, in the NLP context, there are these sort of decoding schemes where you effectively, you're generating a probability distribution, you just sample from that, and then you end up with discrete structures, and then you're effectively doing some sort of ensemble matching between the generated structures and the original one. So that's certainly another approach. And if one were to want to push this approach in the direction of trying to predict ensembles of proteins, which makes sense because proteins are not rigid objects, I think that's a natural thing to do in that context.
One more. Yeah, maybe it's too much, but you opened a very interesting conceptual discussion at the end.
Yeah.
Could you just expand a tiny bit more on the concept of formalizing priors? What's a good example to make that clear of what that is?
Yeah.
Perhaps we take that offline, but maybe.
Yeah. Well, so in a previous life, I spent a few years on trying to use kind of Bayesian non-parametric approaches to do this sort of thing. So they're very rich structure, again, actually from the NLP literature, that tries to kind of capture priors of what natural languages at least look like. I mean, going from kind of Chomskyan stuff all the way down to kind of more modern constructions. And that, I think, is sort of generally a sort of a worthwhile space to explore. I spent about 4 years trying to make it work. I couldn't really make it work because the computational just sort of requirements were too expensive. This is 6-7 years ago. I think now things have changed quite a bit. So it may be worth revisiting actually that space. Yeah.
Let's thank our speakers again.